Hello everyone, welcome to a new episode of Prelims Warm Up. This initiative has been brought to you by Aptiplus Academy for Civil Services, and my name is Shaik Datta. I'm the faculty for Geography and Environment at Aptiplus Academy for Civil Services. So as you might be aware, in this initiative, every day we are discussing 5 questions which had appeared in the previous year UPSC Prelims examination. So today also we have a session where we are going to discuss 5 such questions which were asked in the previous year UPSC prelims examination. So let us start and see the first question for today. Now the first question that we have is from Science and Tech. Okay, from Science and Tech, and this question was asked in the year 2019 prelims of UPSC. So this question is in the context of wearable technology. So the question is asking, okay, in the context of wearable technology, which of the following tasks is or are accomplished by the wearable devices? So in the context of wearable technology, which of the following tasks is or are accomplished by wearable devices? Number 1, it helps in location identification of a person. Does it do so? Yes. Number 2, sleep monitoring of a person. So do wearable devices do so? Yeah, that is also correct. The third statement which has been given is assisting the hearing impaired persons. Okay, those people who are not able to hear properly. So assisting the hearing impaired person. Wearable devices, will that help? Yes, that will also help in such a case. Okay, so you must have seen that people will fit a device in case if they cannot hear properly. Okay, so when we talk about wearable technology or wearable devices, they are capable of helping human being by all these ways or through all these ways. So the right answer should be all the 3, that is D, 1, 2 and 3. So the correct answer is D. And what is the explanation? Let us see. Wearable technology, it is a category of electronic devices that can be worn as accessories, embedded in clothing, implanted in the user's body, or even tattooed on the skin. I'll repeat once again. Wearable technology, it is a category of electronic devices which can be worn as accessories, embedded in the clothing, implanted in the user's body, or even tattooed on the skin. These devices, they are hands-free gadgets with practical uses. They are powered by microprocessors and enhanced with the ability to send and receive data via the internet. Okay, so they are hands-free gadgets. They will have very practical uses, day-to-day uses, applications. They are powered by microprocessors and they are enhanced with the ability to send and receive data through the internet. For example, location and position tracking sensors. Location and position tracking sensors. For example, GPS is there, altimeter, which will measure the altitude, magnetometer, magnetic field, compasses, accelerometers. All of these are examples of location and position tracking sensors. They are the most common type of sensors on wearable devices such as activity trackers, smart watches, or even medical wearables, where they are used to check the physical activity and health of the patients. Okay, where they are used to check the physical activity and the health of the patients. Similarly, we'll also get to see fitness trackers will be there. They are known as activity trackers. They are typically worn on the wrist, on the chest, or the ears, and they are designed to monitor and track outdoor sport activities and measure fitness related metrics. For example, the speed and the distance of running, exhalation, pulse rate, sleeping habits. All of these can be measured with the help of various wearable devices. For example, we have fitness trackers, which you must have seen a lot of people use them when they are doing exercises or even in the gym or even when they are jogging. Okay, so fitness trackers, they are also called as activity trackers, and they are typically worn on the wrist, on the chest, or on the ears, and they are designed to monitor and track outdoor sport activities and measure the fitness related metrics. For example, the speed and distance of running, exhalation, that is giving out of breath, that is exhalation, pulse rate, and sleeping habits. Also, we get to see that in the recent times there has been a surge, a new surge in wearable technologies, especially those which are worn on the ear, and they are referred to as hearables. And it is not just people who are disabled or people who cannot hear properly. Not only they wear such kind of devices on their ears, but we also get to see that normal people also will be wearing various devices on the ears. For example, we also use earphones, right? Earphones, earpods, and all that. So these are also examples of wearable devices. So the correct answer is D, all the 3. Here also we have a little bit over here, hearing aids, that is also a category of hearables. It involves the use of microphone, a speaker and an amplifier. It amplifies the sounds to help people with hearing impairment to hear. Okay, those people who cannot hear properly, they can use these hearing aids which will enable them to hear. It will amplify the sounds, so it becomes easier for the person to hear. An example has been provided, but the main thing is that all the three are ways in which wearable devices they can be beneficial to human beings. Moving on to the next question that is there, and next question has also been asked in the 2019 UPSC prelims, and this question is also from Science and Tech. So this question, it is asking about RNA interference technology, or simply called as RNAi technology. So RNA interference technology or RNAi technology, it has gained popularity in the last few years, and why? Okay, RNA interference technology, it has gained popularity in the last few years. Question is why? So you have four statements. Number one, it is used in developing gene silencing therapies. It is used in developing gene silencing therapies. Number two, it can be used in developing therapies for the treatment of cancer. Number three, it can be used to develop hormone replacement therapies. And number four, it can be used to produce crop plants that are resistant to viral pathogens. Okay, so you have got four statements, and you have been asked, okay, how does RNA interference technology or RNAi technology, how and why it has gained popularity in the last few years? Number one, it is used in developing gene silencing therapies. Number two, it can be used in developing therapies for the treatment of cancer. Number three, it can be used to develop hormone replacement therapies. And number four, it can be used to produce crop plants which are resistant to viral pathogens. So let us first go through the explanation part of this. Now, what is RNAi? It is a gene silencing technology. What does it do? It inhibits protein synthesis in the target cells by using double-stranded RNA. Okay, first thing you remember, it is a gene silencing technology. So where was gene silencing technology? First statement. So first statement becomes correct. So this statement is definitely correct. Let us look at the second statement which is there. This RNAi technology or RNA interference technology, it has huge significance within the Indian context, considering the deep-seated resistance over the years to Bt cotton. Okay, Bt cotton, it is a type of bacteria or soil bacteria has been inserted in this cotton. So we get to see, considering the deep-seated resistance over the years to Bt cotton and other genetically modified seeds. So RNA reliant solutions, they can be a viable alternative to this genetically modified seeds. Okay, so RNAi has huge significance within the Indian context due to the huge amount of opposition which has been faced by various genetically modified crops and seeds, for example, like Bt cotton. And what can be done is that instead of using such genetically modified seeds, RNA reliant solutions can be a good alternative to them. So RNAi is used in functional genomics. What is meant by functional genomics? The systematic analysis of the loss of function phenotypes induced by RNAi triggers, and developing therapies for the treatment of viral infection, dominant disorders, neurological disorders, and many types of cancers. Okay. How in vivo inactivation of gene products linked to human disease progression and pathology. Now, it is not necessary that you have to go so deep into this particular technology, because it is not possible for a general candidate to be aware about these terms, and that is also not what UPSC is expecting. Why this question was asked? Because it was there in the current affairs of that particular year. But over here, we get to see that, first of all, it is used in the gene silencing therapy. Is it correct? Yes. Can it be used in the in developing therapies for the treatment of cancer? So right now we have seen that it helps in developing, or it can be used in developing therapies for the treatment of viral infections, dominant disorders, neurological disorders, and also many types of cancer. So of course you can understand that can be used in developing therapies for the treatment of cancer. This statement is also correct automatically. But can it be used to develop hormone replacement therapies? I do not think we have ever discussed about hormone replacement therapy as such. So no, RNAi, that is RNA interference technology, it cannot be used as an alternative, or it cannot be used for hormone replacement therapies. On the other hand, we saw that it can be used to ensure some solutions of viral, for example, can be used for treatment of viral infections. So here also we get to see viral infections are not only confined or limited to human beings, but viral infections can also affect plants as well. So in the fourth statement, we get to see that it has been mentioned that this particular technology, RNAi technology, it can be used to produce crop plants that are resistant to viral pathogens. So automatically this also becomes correct. So our correct answer is A, 1, 2, and 4. Okay. Correct answer is A, 1, 2, and 4. Moving on to question number 3. And this question is also from Science and Tech. And this was also asked in the 2019 UPSC Prelims. So the third question is asking, okay, recently scientists observed that the merger of giant black holes billions of light years away from the Earth. Okay, recently scientists have observed the merger of giant black holes billions of light years away from the Earth, and what is the significance of this observation? Okay, how the merger of these giant black holes, which are very far away from the Earth, and their observation of this merger, how is that relevant? How does that help? What is the significance? A. Higgs boson particles were detected. Okay, Higgs boson particles were detected. B. Gravitational waves were detected. C. Possibility of intergalactic space travel through wormhole was confirmed. And D. It enabled the scientists to understand singularity. Again, this question was asked because it was there in the news. And that is why, again, for a normal candidate, if it has not been in the news, you are not supposed to know this as well. So let us look at the explanation for this particular question. Okay, how the observation, or what is the significance of the observation and of the merger of giant black holes which are very, very far away from the Earth. So if you look at the LIGO detectors, it discovered the first gravitational waves which are produced by two giant merging black holes in 2016. Okay, so you had the LIGO detectors which were able to discover the very first gravitational waves which are produced by two giant merging black holes in 2016, and hence we get to see that the 2017 Nobel Prize for Physics, the 2017 Nobel Prize for Physics has been awarded to the LIGO Virgo collaboration for their detection of gravitational waves arising from the merger of the two black holes. So that is the reason why this particular question was asked, that we get to see the 2017 Nobel Prize for Physics. It has been awarded to the LIGO Virgo collaboration for their detection of gravitational waves which are arising due to the merger of two giant black holes. So the correct answer is B. Okay, so the correct answer for this is B. Moving on to the fourth question, and this fourth question was also asked in UPSC Prelims 2019, and this question is also from Science and Tech. The question is asking, which of the following are the reasons for the occurrence of multi-drug resistance in microbial pathogens in India? I'll repeat once again, which of the following are the reasons for the occurrence, which of the following are the reasons for the occurrence of multi-drug resistance in microbial pathogens in India? There are four statements which have been given. Number 1, genetic predisposition of some people. Number 2, taking incorrect doses of antibiotics to cure diseases. Number 3, using antibiotics in livestock farming. And number 4, multiple chronic diseases in some people. So the question is asking, okay, which of the following are the reasons for the occurrence of multi-drug resistance in microbial pathogens in India? Number 1, genetic predisposition of some people. Number 2, taking incorrect doses of antibiotics to cure diseases. Number 3, using antibiotics in livestock farming. And number 4, multiple chronic diseases in some people. So this is something that we get to see happening quite frequently in case of India, that is, we get to see that many of the microbes, they have developed resistance against many of the drugs, and such kind of microbes or such kind of bacteria, they are called as multi-drug resistant bacteria or microbes. Okay, so let us see the explanation for this particular question. First of all, antibiotic consumption in human beings. Antibiotic consumption in human beings, if we see, In 2014, India was the highest consumer of antibiotics, followed by China and USA. So in 2014, we get to see that India was the highest consumer of antibiotics, followed by China and the United States. In India, antibiotic FDC, what is the full form for FDC? FDC stands for fixed dose combinations. So in India, antibiotic FDCs or fixed dose combinations are heavily prescribed even without the knowledge of a proven advantage over single compounds. What is meant by FDC? So I've just given over here also the definition for FDC. FDC or fixed dose combinations are pharmaceutical products, for example, tablets, for example, ointments, or even suspensions where two or more drugs are combined in one single product. Okay, so in India, we get to see even for small diseases, we tend to pop a lot of pills or we tend to have a lot of ointments. And the problem is that in these pills, in these tablets, in these capsules and ointments, there are more than one compounds which are there. Okay, so what happens is this: these are called as antibiotic FDCs or fixed dose combinations. These are heavily prescribed even without the knowledge of a proven advantage over single compounds. Fixed dose combinations are pharmaceutical products such as tablets, such as ointments, suspensions, etc., where two or more drugs are combined in one single product. What happens is that if there is injudicious use, that is excessive use or use without proper prescriptions, if injudicious use of antibiotic FDCs are there, it could lead to emergence of bacterial strains which are resistant to multiple antibiotics. However, the per capita consumption of antibiotics in India, it is much lower than in several other high income. Per capita अगर देखा जाए तो per capita it is much lower. But on the whole अगर देखा जाए, then we get to see that India was the highest consumer of antibiotics. उसके बाद आता है China, फिर जाके we have USA. Now, antibiotic consumption in animals. The use of antibiotics in food. Animals play a major role in human health, as antibiotic-resistant bacteria can be transmitted between humans and animals through contact in food products and from the environment as well. So, basic thing is this: okay, antibiotics in India, if we see, we use antibiotics excessively, sometimes even without proper reasons, without proper prescription. You'll have a lot of over-the-counter medicines where we have more than one antibiotic combinations, which are called as antibiotic FDCs, and these we use to treat very minor ailments. Ultimately, what happens? We become resistant, or these microbes and all, they also become resistant to these antibiotics, and later on, when later on, it is possible that these antibiotics will not work any longer on our bodies. And it is also possible for us or for these microbes to become resistant to antibiotics, because a lot of such antibiotics are not only fed to human beings directly, but also they are given to livestock, to various animals, which are consumed by human beings later on. So that is how you must be aware about the terms like biomagnification, bioaccumulation. So that is how ultimately the antibiotics are able to finally reach the human beings, and that leads later on to development of resistance to antibiotics. So in the question it had asked, okay, which of the following are the reasons for the occurrence of multi-drug resistance in microbial pathogens in India? So out of the following, we get to see genetic predisposition of some people. Is that the reason? Answer is no. Taking incorrect doses of antibiotics to cure diseases, can that be the reason? Yes, that is definitely one of the reasons. And also we saw that not only taking incorrect doses of antibiotics to cure diseases, but also we get to see a lot of antibiotics are fed or injected into livestock, which later on are able to make their way into the bodies of human beings. So both 2 and 3 are correct. Both 2 and 3 are correct. So the correct answer should be B. Okay. So the correct answer is B. Just remember what is meant by antibiotic FDCs, okay, fixed dose combinations. Next, the last question for today. Here we get to see that this question was asked in the year 2019 prelims, and this question was also from Science and Tech. The question is asking, okay, what is Cas9 protein that is often mentioned in news? What is Cas9, Cas9 protein that is often mentioned in the news? A. It is a molecular scissor used in targeted gene editing. Cas9 protein. What is it? It is a molecular scissors which are used in targeted gene editing. B. It is a biosensor used in the accurate detection of pathogens in patients. C. It is a gene that makes plants pest resistant. And D. It is a herbicidal substance synthesized in genetically modified crops. Okay. So out of this, which is the correct answer? Cas9 protein was there in the news. It has been in the news for a long period of time. And this question was relatively easier compared to the previous ones. So the right answer should be A. That is, it is an example of this one. It is an example of a molecular scissor which is used in targeted gene editing. It is like a molecular scissor which is used in targeted gene editing. So let us quickly go to the explanation part of Cas9 protein. Cas9, also called as CRISPR-Cas9, CRISPR, CRISPR-Cas9, it is a system which is used by bacterial cells to recognize and destroy viral DNA as a form of adaptive immunity. CRISPR-Cas9, it is a system which is used by bacterial cells to recognize and destroy viral DNA as a form of adaptive immunity. Using components of the CRISPR system, researchers can remove, add, or alter specific DNA sequences in the genome of higher organisms. Okay. So this CRISPR-Cas9, it is a system which is used by bacterial cells to recognize and destroy viral DNA as a form of adaptive immunity. Also, we get to see by using components of this CRISPR system, researchers can remove, add, or alter specific DNA sequences in the genome of higher organisms. This gene editing tool, it has two components. There are two components in this gene editing tool, that is CRISPR-Cas9. A single guided RNA which contains a sequence that can bind to DNA, and the Cas9 enzyme which acts as a molecular scissor which can cleave the DNA. Okay? So this CRISPR-Cas9 system, it has two components. Number one, a single guided RNA that contains a sequence which can bind to DNA, and the Cas9 enzyme which acts as a molecular scissor which can cleave the DNA. But I believe all of this might sound a bit like Hebrew to all of you. So basic thing is that, again, don't have to go too much into the details. This is just for your prelims examination. They won't be asking you in such great technical, what is this CRISPR-Cas9 used for? So your right answer is A, a molecular scissor which is used in targeted gene editing. Okay, it is a molecular scissor which is used for targeted gene editing. So with this, we come to the end of today's session of Prelims Warm Up, and hope you have liked this video. In case if you have, please like, please share with your friends who are also preparing for UPSC, and also subscribe to AptiPlus's YouTube channel for more such informative videos. We'll be back at Warm Up. Till then, stay safe and study hard. Thank you so much.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.